your membrane. There is the bone underlying the cartilage that I indicate here. There's the surrounding tendons and ligaments, the muscle. All of those tissues are important. And the reason that I'm focusing on cartilage today is, as I said, I'm most familiar with it, and it's one of the most studied tissues there. But that doesn't mean the others are not important. But overall, similar principles will apply to all of those tissues. So we're going to talk about cartilage and osteoarthritis. And here's another schematic of what's happening to this cartilage and osteoarthritis. And here again, you see in the light blue area is the cartilage that I try to outline here. And one thing that happens in osteoarthritis is that this cartilage gets lost. And then if you look at the other part where it says osteoarthritis or eroded cartilage, you see that all of a sudden this cartilage is gone. There's no more of this blue tissue left, and bone rubs on bone. And that's, of course, part of the problem in osteoarthritis. One of the things that we can observe, not the only outcome, not the only symptom as we know, but one of the major ones, one of the hallmarks that we can follow, that we can see, for example, in X-ray, the joint space narrowing, that we can see when we do tissue sections, when we slice the joint in an animal model, and you're going to see some examples of this later today. Here's now one example where we have cartilage from an osteoarthritis model. What you see on the left here is a tissue section, a slice of cartilage, and all the red that you see is a staining for the matrix that's surrounding the cartilage cells. So the little small roundish areas, I'm circling one, these are the cells of cartilage. We call them chondrocytes, but they are only a small component of the cartilage. So most components of the cartilage is this reddish stained area, which is extracellular matrix. It's made out of collagens that you probably heard about, and other molecules that we know as proteoglycans, glycosamines, and those are the ones that are actually stained red. And then what you can see at the very top here is the surface. This is the joint space where you see that where you would have the synovial fluid that I talked about before. So this is what healthy cartilage looks like. Nice red staining throughout, and then a smooth, very smooth surface. If you now go to the other panel, to the right panel, that is labeled OA for osteoarthritis, you see it looks very differently. You see, first of all, the surface is no longer smooth anymore. It has all kinds of irregularities, it's ruptured, it is in the process of breakdown. The other feature that you see that's very obvious is that there's much less of the red stain. That means much less of these proteoglycans that are very, very important in the functions of cartilage, in the mechanical functions, because cartilage really has a mechanical function in the joint to allow us to locomote and also to maintain those heavy loads when we walk, for example. So you see two changes, as I said, the loss of the smooth surface and also a loss of the red stain, a loss of extracellular matrix. This is what's happening in osteoarthritis, one of the many things that are happening, but in order to understand at the more mechanistic, at the more molecular level, what's going on, and in order to find new ways to address that, new ways to heal or interfere with medication into the osteoarthritis process, you need to understand a little bit more about cartilage. So this is a schematic of the same thing that I've shown you a little bit blown up and as a schematic. That you have down here again in the lower right corner, I'm circling one of those cells, one of these cartilage cells, or we call them chondrocytes. So that's the cell, and that's the cell, that's the one part of the cartilage that is responsible for the biology. So these cells produce everything that's out surrounding the cell, all the components of the cartilage, like the collagen that I talked about, the proteoglycans. All of those molecules are produced by the cells, by the chondrocyte cells, by chondrocytes, the cartilage cells. Each cell has a nucleus, and I'm circling it right now, the cell nucleus, which is where all the genetic events take place. That's where the genes are stored, the chromosomes are in there, and we're going to talk about that later. So the genetic events, they happen mostly in the cell nucleus, and we're going to come back to those events later. Now, as I said before, the chondrocyte is only a small part of the cartilage, surrounded by this vast amount of extracellular matrix. Like you can see here in black, that would be the collagen fibers, very important in cartilage. In blue, you have a molecule, a polymer called hyaluronic acid, and that's been attached to a lot of those proteoglycans that we talked about as well. The main one being called aggrecan, which is one proteoglycan there. There's all these little letters here. All of these are aggrecan molecules, and again, they are very, very important, both the collagens and the aggrecan, for the mechanical functions of the cartilage. Another component that we're showing here are these proteases, like these big green circles. So these are enzymes, proteins, whose function it is to break down other proteins. And in normal, healthy articular cartilage, we need a little bit of this. We need a little bit of turnover, because those molecules, like the collagens, the proteoglycans, they get old, they undergo certain chemical damage. So you need a little bit of turnover to replace the older ones with newer ones that are then produced by the cell again, by the chondrocytes. So those proteases that are quite important, and we come back to them, they are made to have a normal role in normal healthy cartilage, but there is not a whole lot of them. The final component is what I'm circling here right now in the left part, the little pinkish reddish circles. These are called growth factors. So these are other proteins that are made by the cell, by the chondrocyte again, are made by them, are transported out of the cell. They can diffuse through the cartilage, and then they can act on neighboring cells and induce certain biological responses. For example, this growth factor could tell a cell to divide, to make more cells, or could tell the cell to make more matrix, to make more collagen. So these are signaling molecules that are quite important, and we're going to have another example of those as well. All right, but the main point here is that the chondrocytes themselves, the cartilage cells, they make all this extracellular matrix, the collagens, the proteoglycans like aggrecan, and they also make those proteases. And what is usually happening in a healthy joint is that these processes are in balance. So we talk about anabolism. Anabolism is when we produce extracellular matrix. So, and we can call those extracellular matrix molecules like the collagen and the proteoglycan. These are the good guys. These are the ones that are important for cartilage functions. We absolutely need them there. And as I said, those good guys, those anabolic factors, are usually in balance with the catabolic factors. And catabolic factors are, for example, these proteases that we just talked about, these proteins, enzymes that break down the catabolic, that break down the anabolic molecules, that break down the matrix molecules. And as I said, in a healthy joint, we have a balance between anabolism and catabolism. We always have as much matrix production as we have destruction, degradation at any given point. In osteoarthritis, the situation changes, and then all of a sudden, catabolic processes take over, and we have a lot more catabolic processes, a lot more proteases, not enough production of new extracellular matrix molecules, and that's what leads to this cartilage breakdown, which we've seen in the earlier slide, where you see the loss of red stain, and then you see the damage at the cartilage surface. That is when catabolism is stronger than anabolism, when we lose this balance. Now, there's many different things that can trigger this loss of balance. Injuries, for example, are one of them. You can have, let's say, a ligament rupture, other joint damage that can lead to a loss of this balance. Can be obesity is an important factor that can change this balance. But what we know for a number of years now is that genetic factors are extremely important. And this is just a graph that I took from a publication from Tim Spector, where they look at genetic factors in osteoarthritis cases in different joints. So you see the bar graph shows you the percent of cases that are due to genetic factors. And you can see that in some cases, like the cervical spine, the lumbar spine, the hand, 60–70% of osteoarthritis cases are actually primarily due to genetic factors. More than half of them have the cause is genetic and not anything else, which is quite high. In other joints, it's a little bit less. In the hip, it's less, and it's lowest in the knee, which is maybe not surprising. Because the knee is probably the joint that gets hurt most often in sports injuries, and so more of the knee osteoarthritis cases are because of injuries and things like this, as opposed to genetics. So why are those genetic factors so important? What do the genes really do? And they do a lot. They really control many aspects of the joint function and of the cartilage function. For example, they determine the architecture of the joint, and that's extremely important. The joint architecture forms while we are growing, while we're developing, while our bones and cartilage and joints form. If you have a less than optimal architecture of the joint, if the joint is slightly deformed, which you might not notice early on, but if you have it slightly deformed, then the whole biomechanics are different. Then the way that the different bones fit with each other and the forces on the joints are very different. And if they are not optimal, then these factors, these loads can lead to cartilage degeneration on their own. So genetic factors during development control the architecture of the joint. Genes are also important for the strength of the tissues. This is, for example, determined by those collagen and proteoglycan molecules that we've been talking about. How much of them do we have, and do they have the right sequence? Are they properly formed, or do they have slight changes that make them less functional? Genes control the division of cells. How many chondrocytes, how many cartilage cells do we have there? Do we have enough to maintain a healthy cartilage? Can it maybe respond to damage by making more cartilage cells or not? And finally, genes control the function of those cells. And one of the functions, as I said, is to produce enough of the collagens, produce enough of the proteoglycans, and also produce these proteases, the bad guys that break down the matrix. So genes control all of these factors. So I'm going to show you a few slides on genetic information, just to give you a little bit of an introduction. I hope it's not too boring, and I also hope I'm not talking about your head, because I don't really know the level of knowledge you have on genetic information. It's going to be a little bit dry, lots of text the next few slides, and it's not absolutely essential that you understand everything there, but I thought I'd give you a little bit of an overview so you can appreciate what I'm talking about. So genes, genetic information, all the genetic information that we have is stored and also inherited, distributed to our daughters and sons as a sequence of letters. Sequence of letters that we call chemically nucleotides, and that are in a molecule, in a chemical entity that's called DNA, desoxynucleic acid. So this chemical molecule, DNA, is where the genetic information is stored. In humans, we're talking about humans now, this genetic information is 3 billion letters. So it's an enormous amount of information that we have there, 3 billion letters of these nucleotides, and they are distributed over 46 strands of chromosomes of 46 different molecules. And I just jump ahead to the next slide to give you an illustration. So these are the chromosomes in a normal human cell. There's only 23, or actually 24 shown here, because they come in pairs. Most of them come in pairs, the exception being the X and Y chromosome on the lower right hand, which determine our gender, whether we are male or female. But the other 22 chromosomes come in pairs. We have 2 of each. And that's where all our genetic information is stored. You can see they're very different. There are small ones, like the number 21 or 22 here. There are very large ones, number 1 and 2. They are chemically very similar in terms of their components, those letters that make up the genetic information, but macroscopically, the way they look, they are quite different. But that's where all our genetic information, with a very small exception in the mitochondria, which we don't have to worry about today, but that's where all our genetic information is stored. So I'll go back to the last slide and continue there. As I said, the 3 billion letters containing our genetic information. If we take any given 2 humans from anywhere in the world and compare the sequence of those letters, the sequence of all the 3 billion letters, we see that 99.9% of these letters are exactly identical in all of us, whether we are from China, from Africa, from Europe. They are 99.9% of the sequences are completely identical. But there is a small proportion, 0.1%, 1 in 1,000 of these letters are different between 2 people, any non-related 2 people. And these small amount of differences are responsible for many, many differences that we see, whether we are 7 feet tall or 5 feet tall, whether we have blonde hair or black hair, white skin or brown skin. All of these differences are due to genetics, and they are determined by these 0.1% differences. So not only the things that we can really see, not only the differences that we can see are due to those 0.1% of differences. It's actually, for example, how likely we are to get certain diseases, how likely we are to get cardiovascular disease, how likely we are to get cancer, how likely we are to get osteoarthritis, is also determined by these 0.1% of the letters that are different. And the last bit of information that I'm giving you here is right now we don't have the exact number, but it should be around 20,000 of those genes that we have in humans. 20,000 genes, which is quite a large number, also not as large as we had expected a few years ago. And there's another illustration just showing you one of the strands of DNA, this oxynucleic acid, where we have what you can see here in the upper panel. You see these letters. There's actually only 4 letters in the genetic alphabet: A, C, T, and G. And what really determines the function of the gene and everything that's controlled by a gene is the sequence of these letters. So A, C, T has a very different meaning from, for example, if you would read it the other way around, T, C, A. The exact sequence of these letters, these nucleotides, is extremely important for the genetic content, for the information that's stored in those genes. And what we can see down here is then these molecules of DNA. They are highly packaged. They form what's called the double helix. You might have heard about the double helix. So its discovery was one of the really important events in the last century in biology. They are packaged in this double helix, and then they are packaged up at higher levels. So, tell you a little bit more about genes. What these genes, they are really only the carrier of information. They are not the functional molecules. They are not the workhorses. They are not the molecules that do the work. They just contain the information, and that information is then converted through quite complicated biological process into the real molecules that do the work, which are the proteins. So those proteins are the workhorses, but how exactly they look, what they do exactly, that's determined by the genetic information in the genes. The genes contain information for all those proteins, and we already encountered some of those proteins. The collagen is a protein, the proteoglycans, like the aggregan, these are mostly proteins. The proteases, the bad guys that break down the collagen and so on, they are proteins. The growth factors we talked about, all of these are proteins. So they are really the molecules that do the work within our cells, within our tissues, within our body. What's really important is the next concept. So all our tissues within our body have the same genes, the exact same genetic information. Whether you're a brain cell, a liver cell, a cartilage, a bone cell, they all have the exact same genes, the exact same genetic information. But of course, our tissues are different. Our cartilage would work in a brain, and a muscle cell would work in a liver. Those tissues and the cells within those tissues have very, very different jobs within the body. So how come they do different jobs and are good at it when they have the same genetic information? And the answer to this question is that the difference is not in the genetic information, but in how this genetic information is used. So as I told you in the last slide, we have 20,000 genes in our body, and each cell in our body has these 20,000 genes. But in each cell, in each tissue, only a subset of these genes is really active. So maybe 1,000 or maybe 3,000 of those 20,000 genes are active. And the sets that are active in a nerve cell, in a brain cell, are very different from the ones that are active in a liver cell, and they again are very different from the ones that are active in a cartilage cell. So what makes our tissues different is really the patterns of genes that are active, the patterns of genes that produce those proteins at any given time in a given space. So that's a really important concept. All the cells have the same genes, but they are utilizing those genes differently. They're controlling which genes are activated, are switched on, which genes are switched off, how much are they switched on, how much are they switched off. And the same concept then applies to health and disease. So osteoarthritic cartilage is different from healthy, normal articular cartilage. And one of the reasons it's different is that the pattern of gene activity is changing. They have the same genes, but the pattern of gene activity is different. For example, osteoarthritic cartilage, those genes that that are responsible for the proteases, for the bad guys that break down the collagen. Those genes are much more active, much more switched on to a higher level. That's why we all of a sudden have more of those proteases there. And that's why this balance is lost that I talked about, and we're losing matrix, and that leads them to cartilage breakdown. So how does genetics really contribute to osteoarthritis? And there's two major things, two major scenarios, two major models that we can apply. The first one is the difference in nucleotide sequence, the difference in the sequence of these letters that I talked about. I told you 0.1% of those letters are different in any two persons, and those 0.1% differences, they are responsible in making us more or less likely to develop osteoarthritis early in our life or even later in our life. And those differences can make us more likely, for example, because we might have a different letter in a gene for a matrix molecule like collagen 2, and because of this difference, this molecule looks a little bit different. It's maybe a little bit less stable, doesn't last as long, doesn't resist a force on the joint as much. So then this protein is functioning just maybe 1 or 2% less perfect than the other variant. And that means over time, this less perfect function will then make the cartilage weaker, will make it more likely to be degraded and lost, and then we end up with osteoarthritis. So it's these differences in the nucleotides that are important in the nucleotide sequence. And the same differences can also apply to those patterns of tissue of expression. So you can have a difference in the nucleotide sequence that causes us to have less collagen 2, lower levels of collagen because of one of those differences. The second model that I'm talking about down here is that the genetics also control pathways that determine how cells respond to their environment. So all our cells respond to what they see around them. They respond to hormones and growth factors. They respond to biomechanical loads. They respond to cytokines and inflammation. So, and all these responses are genetically determined. For example, if we have a small or major injury in our joint, that changes again how the load that the chondrocytes, the cartilage cells experience. And these loading patterns can then switch on and off some of those genes. For example, what often happens if you have more load on the joint, the proteases again, those bad guys that break down collagen, can be switched on. And again, this is a genetic pathway that determines those responses. So both of these models here are important. Both of these models we have to keep in mind when we talk about genetics and osteoarthritis. So if you go talk about specific connection between genes and osteoarthritis, one of the sadder part is that we don't really know many of the genes that contribute to the development of osteoarthritis. And that is something that probably as a patient or patient advocate, you know that we, as researchers know, osteoarthritis is just not a disease that gets all the attention it deserves. It just doesn't get the same attention that cardiovascular disease or diabetes or others get, which we think is not deserved. But generally, there's less funding for osteoarthritis research worldwide, and that means the research lags a little behind other fields that have 10 or 20 times the amount of funding to do this research. So most of those genes we don't really know yet, and we are working on this. But what we know so far is that different genes appear to contribute to osteoarthritis at different sites. There's different genes involved in knee osteoarthritis compared to hand osteoarthritis. The different genes involved in males versus females and so on. What we also know, like for many of the common diseases, including cardiovascular disease, is that it's not just one gene per patient that's making them more likely to develop osteoarthritis. It's a combination of many genes, which we call a multifactorial disease. And another important concept is that these genetic effects, they often work together with environmental factors to determine our risk of osteoarthritis. So if you have a genetic factor that makes you more likely to develop osteoarthritis, this might not give you osteoarthritis on its own, but it's osteoarthritis, for example, with the wrong patterns of exercise, osteoarthritis together with obesity, osteoarthritis together with some genetics together with some injuries. So all those factors work together. It's not just a genetic effect. You can have a genetic, what we call predisposition, a genetic factor that makes you more likely to develop osteoarthritis, but that doesn't mean you necessarily have to get osteoarthritis if you live healthy otherwise. If you, for example, stay lean, do the right amount of exercise, muscle strengthening and so on, you can work against this genetic risk that you might have. That's a very important point. So genetics is not just, you have that gene, you get a disease. It's not like this. There's ways to overcome that. So what are the current approaches in research? One of the big ones is involved in finding all those genetic variations between different humans that determine how likely we are to develop osteoarthritis. How likely is it that we're going to get osteoarthritis sometime in our life? So we have to identify those differences between different humans. So in those 0.1% differences, which ones of them are linked to osteoarthritis, which ones are no. The second big area that we're working on is to identify the changes in patterns of gene activity in osteoarthritis. As I told you, for example, those protease genes, those genes involved in the catabolic functions, they often become more active in osteoarthritis. But we haven't identified all of those changes. We have to find more of them to figure out what is going on. Where can we get a handle on, for example, for improved therapy? And the third major task ahead is not only make a catalog of all those different changes in points 1 and 2, but also test whether they're really important, because you might have some changes that seem to be associated with osteoarthritis, but in the end, no, they're just some bystander effects. So you have to really functionally test whatever changes you identify in 1 or 2. You have them test them in 3, whether they're really related to osteoarthritis, whether they're really important. And the way you usually do this is using animal models. Altogether, those three tasks will actually allow us to identify novel strategies. First of all, to prevent osteoarthritis. That must be the ultimate goal in the end, to prevent the first occurrence of arthritis. But then next step will be to diagnose it better. You probably know osteoarthritis diagnostic is not very well advanced these days. It can only be diagnosed at a late stage. You'd like to diagnose it more early, more easily than with an X-ray or something like this. And ultimately, we want to use that information to find new therapies, of course, to find new therapies to make to stop osteoarthritis, and maybe even reverse it, heal it, to overcome it. And all of these studies will involve genetic approaches, are targeted at genetics, and that was the main message. I had some more slides to give you an example from my own work, but I think we're pretty close to the end of the talk, so I will just switch off my talk and ask Dana for a minute whether she wants me to continue or wants me to come to the end now.
Frank, you can feel free to continue if you still have a few more slides to go. We have time.
People still awake?
They're still awake.
Okay, then I'll continue for a few more minutes. I can go through that rather fast. Okay, so for the last few minutes, I'm going to give you an example from our own research that shows you how we go about that. And as I just said, we usually have to do those studies in animals for a number of reasons. First of all, we want to catch osteoarthritis early. We want to diagnose it early. We want to treat it early before it gets really severe and with severe limitation. So that's why we have to do it in rats, because we don't really have human subjects where we can get early cartilage, where we can do ethical experiments trying to heal cartilage in a living human. It's just not possible and not ethical to do that in humans. So we usually use animals, in our case mostly rats and mice. I'm going to talk about rats right now, where we we induce surgery in the knee of those rats. The surgery induces osteoarthritis, and on top of that, we put them on these rotating cylinders where they are forced to walk and extend their joints, a little bit like stair climbing or so they cannot rest them. And we have shown in the past that leads to something in the rats that's very similar to human osteoarthritis. And you can see here in the tissue section again, same thing as I showed before. In the on the left hand, you see that what's called sham, so it's our control that does not have osteoarthritis. You see a nice smooth surface of the cartilage. You see a nice red stain below for the cartilage matrix. If we induce osteoarthritis on the right, you see again the surface is no longer smooth. There's lots of damage up here. The cartilage is beginning to erode, to go away. And what you also see at that surface, the red staining is gone. It's only that greenish staining left. So the extracellular matrix is being lost, and that's the first stage in osteoarthritis. So what we have used this model for is what we call a gene chip or microarray study. And these are relatively novel tools that we can use in genetic research, where we can have these chips. They are pretty small. They are a few square centimeters altogether, and they have probes on them where we can look at the activity of basically all the genes in a rat genome, or we could do the same in the human, but as I said, we're not doing that in humans at the moment. But in a rat genome, we can look at every gene in the rat to see how active it is. And if you look at the picture at the bottom, you can see in the top left corner, there are 3 very bright dots. That means those genes that we are probing for with those white box, they are very, very active. There's lots. They are turned on highly. That's why we get a very bright dot. And then you have other areas like down here in the bottom right one, where it's very, very dark. Those genes are switched off and not on. So we can use these gene chips or microarrays and then take the cartilage from our operated rats, compare them to our rats that don't have osteoarthritis, and see which genes change in the cartilage in osteoarthritis compared to control. And we have done that study, and we have shown that actually, if you just look at the left bars, if we look and just look at the black bar, which is the osteoarthritis cartilage compared to control cartilage, there's almost 1,000 genes that are more active in osteoarthritis. We call them upregulated. And there's more than 600, 700 genes that are less active in osteoarthritis. So out of the 20,000 genes that the rat has, very similar to us genetically, out of these 20,000 genes, there's about 1,600, 1,700 that become either switched on more or switched off more. There's a lot of changes, a lot of targets to look at. And the important thing is that maybe 100 of those genes have been implicated in osteoarthritis before. 100 of those genes we knew were involved in osteoarthritis, which is a good control because we find the known players in our model. But then there's another 1,500, 1,600 new genes that seem to be involved, but nobody has really looked at them. There's a big field for us to study to find new treatments in the end for osteoarthritis. And I just show you an example of one of those factors that we then focused on, which is called TGF-alpha, or transforming growth factor alpha. It's one of the growth factors that I talked about before. It's produced in cells, it's exported out of the cells, diffuses through the cartilage, and then it can act on other cells and influence what they are doing. If we then look over here in sham, is again our control, our healthy cartilage, we don't see a lot of brown staining for this growth factor. There isn't much of it there. But if you then look on the right in our osteoarthritic model, you see there's a lot of brown staining up here, very intense, very strong staining. There's lots of that. And this is exactly there where the cartilage damage occurs. So this factor is not usually there on healthy cartilage, but then large amounts are being made once it gets osteoarthritic. So that suggests this factor is maybe one of the factors that can drive osteoarthritis. And then we did some in vitro studies, so studies in a cell culture system, or in this case we took pieces of cartilage from adult rat knees and put them in a culture dish for up to 7 days and treat them either just with control down here, so they have just a normal culture medium that keeps their cartilage healthy, or we took the same medium and add that TGF-alpha, that growth factor, and then we stain for something that's called MMP13. That's one of the proteases, one of the bad guys that breaks down collagen. And then again, we look for the brown stain. You see that if we add TGF-alpha, there's a lot more brown stain than in the control. So it's another indication that not only is TGF-alpha found more, it's not only activated in osteoarthritis, if we add it, it can also promote some molecular events that are associated with osteoarthritis. So it's another very good indication that is really an important factor. And to ultimately prove it, we went back into our rat model and used a drug that is known mostly for the cancer research that can block the TGF-alpha signaling. So that drug blocks the activity, blocks how this growth factor talks to other cells. And we induced osteoarthritis in our rats again with the same surgery as before. And you can see over time, on top is 4 weeks, then 7 weeks, and down here 10 weeks after the surgery. Those rats get really bad osteoarthritis. You can, maybe let's focus on the 7 weeks. You can see the rat stain is lost if you compare to a control that's healthy cartilage on the left. So that rat stain is lost. And I hope you can also see that, again, the cartilage surface becomes quite irregular. There's little damage. It's not longer as smooth as it used to be. So again, as before, we induced osteoarthritis in these rats. If we then go to the very right column, where we induce osteoarthritis, and at the same point, we give them the drug that blocks TGF-alpha signaling, we can see, again, at the 7 weeks, the cartilage does not look completely normal. It does not look as nice as the one in the sham on the very left, but it looks a lot healthier, a lot better than the one that just has osteoarthritis. There's more red throughout the cartilage, and the surface is a lot smoother. So what that tells us, if we inhibit TGF-alpha signaling here, then we slow down osteoarthritis progression. It doesn't really stop it completely, but it slows it down. And we can also use a scoring system that people have developed to quantify, put numbers on the severity of the osteoarthritis. And if you look at the green at the bottom, that's our control. This is just normal, healthy joints that get tiny little bit of arthritis over time, just with aging. If you then look at our osteoarthritic samples and the blue line on top, they get severe osteoarthritis quite rapidly at 7 weeks or even worse at 10 weeks. And then if you look at our osteoarthritis model that has gotten the drug in red, you see it somewhere in the middle. So definitely this drug is beneficial. It slows down osteoarthritis progression. It is particularly slows these early phases quite a bit. So that might be something that has not been tested in osteoarthritis in humans, but we have now some leads that it could be efficient, especially early on in osteoarthritis. So maybe a new drug that could be tested in the future. And this drug, this lead, came directly out of genetic studies that we have done in our lab and that are going on in many labs at the world at this time. So what are the next steps for this process? Of course, we have to take the step for TGF-alpha from the rat to the human. We know in human osteoarthritis, TGF-alpha is more active, there's more of it, so that fits, but we haven't really tested whether blockade, inhibition of TGF-alpha signaling works there. We have to understand how does TGF-alpha work in cartilage? What does it really do? Is there other factors that work with TGF-alpha that we could block as a new therapeutic target? And similar in my lab and in many other labs in the world, TGF-alpha is only one out of many other genes that we could look at. There are studies like this going on all over the world, looking at other genes that, for example, came from our gene chip studies or from other studies in different labs. So all of these genes have to be looked at in a similar way. So there's a huge task ahead, but on the other hand, there's also a lot of promise that we can find a few genes that are really important that we can develop drugs against that might be able to help us to stop osteoarthritis. And the most important slide is the thank you slide. So in my lab, there's been a number of graduate students, Tom Appleton, Shireen Osmani, and now Michael Pest, who are working on this project. He's done all the work because I'm not in the lab myself. We also have a very good technician, Vajek Pitelka, who's been working with the rats, did the surgeries and so on. We had very good funding from the Canadian Institutes of Health Research, the Arthritis Society, and the Canadian Arthritis Network. And finally, thank you very much to all of you for listening and sticking around and not falling asleep. Thanks a lot.
Thank you very much, Frank. We're going to, Frank, unclick your talk button. We're going to move on to questions now. So if anybody has any questions, you can use the hand raise button. And when you go to ask your question, just press the control key on your keyboard. That will activate the talk button. And make sure you hold it down when you ask your question. If you don't want to ask your questions verbally, you can definitely type it into the chat area as well. Okay, so let's just see if we have any questions. Please use your talk button if you do have any questions, or type your question, use your hand raise button if you have any questions, or type your question into the talk area. Frank, how much time do you have?
I have another 20 minutes or so at least, but I don't see any questions right now.
Okay, so if we don't have any more questions, that's fine. Frank, maybe you could just put your first slide up again, and then if anybody wants to send you an email if they think of a question while they're laying in bed tonight, they can do that. That would be great. And I just want to thank you, Frank. Oh, Claire, do you have a question?
Yeah, there's one question now. Claire, you want to go ahead and ask the question?
Go ahead, Clara. You can ask your question. Clara, are you there? Do you have a question you want to ask Frank? Well, Frank, we know where to find you and we know where to find Clara. So if she needs to ask you a question, oh, okay. Sorry, Clara. Clara, we can't hear you, so perhaps you could type your question into the chat area.
Okay. In theory, yes. We don't know yet whether the pathway that I talked about is really the best target, but in theory, that is an approach that we should and can use. It's a similar approach has been used, for example, in rheumatoid arthritis with the TNF alpha story, if you're familiar with that, where we have block one of those pathways, block what's called a receptor. It's the molecule on the cell surface that receives that message. We can block that. So there are approaches. What makes osteoarthritis more challenging than some other diseases is that you might have to use the same approach for 20 or 30 years in a row, because it's a disease that can start early on and you might have to prevent it for a really, really long time. So that is a challenge in osteoarthritis. But we might come to the point where we know, for example, we have an injury, a joint injury, which is very likely to result in osteoarthritis. Maybe we find pathways where we can block some of those effects early on for the first few weeks after the injury, and thereby reduce the risk considerably and then stop the drug. But it's still early days, and we have to go ultimately to humans to really answer that question.
Frank, it looks like Carrie has another question. If you can read that in the chat
Yeah.
area.
Carrie asks, which is worse, eroded cartilage or regular cartilage? I think it's a continuum. If the cartilage is completely gone, it's worse than having just some of these surface damages. But it's continuing. Usually, it starts with a very smooth surface, and then you get the first irregularities, and then they go deeper and deeper into the cartilage until, in the end, you lose cartilage completely. So it's a continuum. One leads to the other. Does this answer the question? So there's another question from Alexandra. Stem cells are one way that lots of groups in the world are working on to use those stem cells. One of the problems with articular cartilage is once it's damaged, it usually doesn't heal itself. There's very little self-regeneration. It doesn't heal itself. So the ultimate way, the ultimate goal, I think, is to prevent the damage in the first place. I think that should be the ultimate strategy. But meanwhile, yes, we're looking for strategies to replace damaged cartilage, and stem cells is certainly one approach that many people use and that might have promised, but to my knowledge, it does not work yet very well. One of the problems is that you need to have the tissue, the stem cells that you put in, has to integrate very well with the existing cartilage, with whatever's left of the existing cartilage. And that's one of the challenges that need to be overcome. The other problem is you have to make those cells do exactly what you want them to do and become exactly the chondrocytes with the profile of gene activities that you want them to be, and that's hard to control. Sometimes those cells make something else that you don't want to, and you would only make the situation worse. I'm going back to Carrie's question. So she wonders whether it is best she sees something in the slide, and she's correct that there's some kind of repair process going on in our rat studies where it looks like it's an overproduction of cartilage, but what's happening there is really the joint tries to repair it, and that's something that's really not working at all. So this at the 10 weeks, this is worse than before because the repair tissue doesn't work at all, and it's not doing its job, and it will not heal. And I see another question from Clara about the physical activity. I think physical activity is important, but it has to be the right one. So if you have already severely damaged cartilage, if you have some kind of osteoarthritis, running a marathon is probably not a good idea. Doing some other types of activity, strengthening your muscles around the joint is definitely can be beneficial. It has to be done with the right instruction. I think we still don't know absolutely sure what kind of exercise is good, what kind of exercise is bad. So there's still a lot of research going on. But activity is good. What's also important is to keep the weight down, and activity, of course, helps with that. So, but it has to be controlled. It's not just activity per se. You have to do the right kind of activity, ideally with instruction from a professional.
Are there any more questions from the group? Okay, Frank, thank you very much for that very informative presentation. It's great that we were able to start off our series with a presentation about genes, and you were not boring to listen to, contrary to what you thought. I would like to remind everybody in the room that we do have another webinar coming up. It will be on September the 27th at 1 o'clock, and it's with Dr. Bert Taylor, who used to work at the University of Western Ontario, and now he's based in Manitoba at the University of Manitoba. And he'll be talking about physical activity, aging, and arthritis. So if you want to sign up for that, you just keep an eye on our homepage. I'll post the link in the chat area if you don't know our homepage, and I will put the information about registering for that webinar up in September. And also, I will put the information about the archive for those of you that weren't able to watch the whole presentation, or maybe you have friends who weren't able to log in for whatever reason. You can let them know that our presentations will be archived on our website. I think that's it for now. So if anybody has any questions, you have Frank Beier's email address up there on your computer right now, and you can also send me an email, and I will post my email address in the chat area as well. Thank you very much, everybody, for attending today.
Thanks, Dana.